and Masters from Naipan, Hyderabad. He completed his PhD from Naipan, Mohali. Sir is well trained in many sophisticated instruments like flash chromatography, etc. Also, he has many publications in international peer-reviewed journals and presented his work in many prestigious national and international conferences. Sir, now I request you to interact with our audience. Over to you, sir.
Yeah, thank you very much for nice introduction. Uh, First of all, I would like to thank ISF College management for inviting me for this workshop and lecture. And also I would like to thank Dr. Kalichan Sharma, who has contacted me for the conference, and he gave me opportunity to present some of the work or some of the presentation. So I won't take most of the time, because right now in Texas it's already 12 night. So I have to go for the lab tomorrow morning. So I will start my presentation. So is it my screen visible to all of you?
Yes, sir. Visible.
Okay. So as the theme of the conference was the natural product based drug discovery, so I have chosen the topic, the role of natural products in the new drug development or new drug discovery. So as we know, the natural products itself is a vast resources for the development of many medicines or many active molecules. And many of these medicines, today's medicines which we are taking for the particular disease or particular conditions, are inspired from the natural product. Either it is from directly from the natural source, or it has been inspired, or it has been developed or derivatized from the natural source. So the development of drugs from the natural source is a very tedious process, or very we can say lengthy process. It takes a lot of time, lot of effort to develop any lead molecules or drugs from the natural product. So it starts with the plant collection or collection of any product from natural source, whether it will be the from plant source or marine source or animal source. So the first step is the collection of plant materials or animal things or marine source. Once you have your plant collect, then you have to do the extraction, so that you can extract the active molecules or active ingredients from the plant part.
Sorry for
So
interrupting. Sir, can you please go to slide
so you have to extract the different extraction part, like alcoholic part, water extraction, or hexane extract, or different part you have to form the extraction. Once you have extraction, then you need to do the isolation and purification, because the impure compound can do the many side effects, which is not beneficial for the particular effect. So once you have your isolated product or purified product, then you need to go for the bioassay for particular disease.
Excuse
So
me, sir.
based on, yeah,
Sir, your
yes,
presentation is not on full screen. Our slides are not moving. Sir, please present again.
so it is first slide.
Slide, sir.
I'm on the
Uh,
first slide.
Sir, it is not on sharing mode. Sir, slide so kindly so slide. Ji, sir. Please,
Now
sir.
it's coming.
Yes, 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 sir.
Now it's there.
No, 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 sir.
Is it now?
No, no, sir. Kindly take it on sharing mode.
Yeah, it's in sharing mode.
Oh,
I am sharing again.
yes, sir. Yes.
computational tool, that ADME parameter should match with the your in vitro or animal models or in vivo ADME. Then only once like it can correlate your computational ADME to the like animal model ADME. So once you have done all the studies, so it till now it is the like called as the preclinical study. So once you have done all the study up to the preclinical study, then you have to go for the like clinical trials, which has another it takes like from mostly 6 to 10 years, even up to 20 years to develop a drug. So as we saw that it is a lengthy process, also it is a costly process. So to develop any new drug, it takes almost like 15 to 20 years, and also it costs, as per the 2022 estimation, 2,000 US million dollar is required to develop any new medicine. So we can see how costly and how lengthy is the process of development of any new molecules or any new drugs. So as we can see, it is a very costly and very tedious process. You have to be very careful when you are choosing any area of research, when you are doing really a new drug development research. So you have to have very careful to choose a disease, and once you have chosen your disease area, then you need to choose the target. Once you have your target, then you have to identify your bioassay, which bioassay is suitable for the your chosen disease or chosen area of the interest. Once you have completed with the bioassay, then you should have your lead compounds or lead agent, which can be act as template for the discovery of new molecules or new medicines. So once you have your lead molecules, that lead molecules should be purified or isolated, because you cannot take medicine as a whole. You have to have defined what particular moiety or particular compound is responsible for the effect of that particular which is responsible for to treat that particular disease. So once you have your pure compound, you need to do the structural determination by using different sophisticated instrument like HRMS, NMR, or XRD, even MALDI. So you need to do the structural determination of your purified compound. Once you all will complete, like we all will complete these steps, then you can have your medicine. We can say that herbal medicine or natural product based medicine. So how any pharmaceutical companies or any like agency are choosing the drugs? So most of the pharmaceutical companies choosing the drugs based on the disease affecting mostly the higher economic countries, because they need to have some benefits from the like development, because it costs a lot of money, and they have to invest a lot of money for the development of any molecules. Then in returns they need some benefits. So basically most of the pharmaceutical companies will focus on the disease condition which affects the higher economic countries. They will not focus on the countries which affect the lower economic countries. So for example, I have mentioned some of the drugs, so you can saw how much costly these drugs are. There is a drugs which called as Lenmeldi. For one like dose, you need to have 4.25 million like dollar for one dose of the medicine. So we can see how many of us, even how many people in the world can afford these medicines, which is very costly. So that's why companies are very like crucial to choosing the disease or to choosing that like area of research, which area they can choose. So mostly you can say nowadays the most of the research area either as cancer, cardiovascular disease, depression, diabetes, because these are the diseases which affecting either the developed countries or developing countries. So most of the companies are focusing on these area so that he can benefit it from the that particular developing or developed countries. So there are some neglected disease which you can see either no companies or very few companies are focusing on these areas, which includes dengue, mostly affecting the like southern Asian part, including India, and Kalajar, which mostly affect the it shows that 18% of population is affected by the Kalajar in 2020, and leprosy. So in that case, some of the government agency comes forward to invest money to these areas. Even WHO comes forward to invest the money to these areas so that these area can also be taken care. So once you have your area of target or area, you have like area of research, you have chosen the target, then you should able to identify the target where agonist or antagonist will be useful for your disease area, because you have to establish the bioassay for your disease condition. So what is the agonist? Agonist is the condition when any molecules interact with the target or receptors, it enhances the efficacy or it enhances the uh, response of that particular disease, response for the like what we need. And what is antagonist? It antagonize the effect once it contacts with the receptors. So once you are able to identify the target, then you should or we should able to choose the uh, suitable bioassay. What it does mean bioassay? If we we are focusing on if we are focusing on the microbial area, then we should able to do some of the microbial assay, including like cell culture, like antifungal, antibacterial. If we are choosing the anti-cancer area, then we will should able to some do some uh, uh, what can I say? If it is anti-cancer, then we can able to say some of the cell cycle analysis or like cell-based assay. So once you have done the in vitro assay, then you should able to correlate that in vitro result with the in vivo study. So and once you will able to like complete the in vivo study, then also we have to do the ex vivo study. So what is the difference between in vivo and ex vivo study? So in vivo we are seeing the effect inside the animal, and in ex vivo we are taking particular part of the disease like liver, lung, and we are taking outside from the animals, and we are seeing the effect of our drugs. So once we are able to find the bioassay, then we should able to finding our leads. So what are the leads, or how we can find our leads? So by screening natural products, what we have seen, and by screening the synthetic banks, like our already synthesized molecules, what we have, we can also find the lead from there, and by existing drugs. So nowadays, like repurposing of drugs is one of the major area of research where people are repurposing the drugs which already available in the future, but they are uh, using it for the different disease condition by doing some more research. And by using the side effect of some of the drugs, the one of the example for that is minoxidil. So actually the minoxidil has been developed to treat the blood pressure, which is taken as the oral medication. But nowadays it is used as a treatment for hair loss, because minoxidil has the side effect of uh, it grows the hair growth. So that's why nowadays in every formulation which is used for the treatment of hair loss, they use minoxidil as their agent. And also we can find our lead by designing some of the commercial aided drug design or by the serendipity. So most of the drug nowadays, like what we see in 90s or 80s, all of them, or I would say 90% of drugs have been discovered with the serendipity. They were not discovered for the particular area. They have been discovered with the serendipity. Then we can design our lead with the based on fragment based lead discovery. So why isolation and purification is important? So we know that thalidomide case, which has been developed in 1950s, which has R isomer as well as S isomers. These drugs has been given to the useful for the morning sickness. So it was given to the mostly the pregnant lady to reduce their morning sickness, because it has the sedative effect. So later it was found that only R enantiomer has the sedative effect, and the S enantiomers has the teratogenic effect, so which lead to the in 1915, almost half of the population in the like babies, they were born with the phocomelia condition, a condition in which babies were born with the deformities in the limbs and hands. So later it was found that only R isomers is the effective and S isomers is the like teratogenic. So by using simple technique like TLC or even chromatography or HRMS, NMR, you will not able to identify whether it is R isomer or S isomer. So for that, you need to do either like derivatization or some of the other technique you have to identify for the differentiation between the R and S. So that's why you need always the pure compound. For that you need to like you should have the HPLC profile that so that it can say. That's why in IP we can say that this compound or this particular assay is 99.9% pure, even though if it is 99.9% pure, so you should able to or we should able to tell what is the 0.1% impurity in that. So if we are giving any assay, then we have to say that. So that's why isolation and purification is the one of the most like important aspect in any drug discovery, either it is natural drug discovery or it is synthetic drug discovery. So it starts with the like conventional column chromatography. Nowadays we have flash chromatography, even more advanced technique like repetitive HPLC, where you can purify like all the tedious any type of molecules you can purify. So once you have pure compound, then you need to able to tell what is that particular compound. For that you need a lot of instrumentation like HRMS, MALDI, NMR, X-ray crystallography, even many more sophisticated instrument you have to tell that what is that particular compound. So once you have identified your structure, like the structure of the compounds, then you can say that is particular compound is responsible for the efficacy of that particular disease condition. So natural products from where we can get from the microbial world, we can get from the plant source, even marine world, even animal source. From any natural source we can get, that is called as a natural product compound or natural product based drug. So one of the like most famous example from plant source is the morphine, which is used as the analgesic, which is obtained from the Papaver somniferum. We have like studied many like from 10th onwards or 12th onwards, we can see that these are the plant source or plant based drug. Then we have quinine from cinchona bark, which is used as the anti-malarial drug, and this is that quinine act as the template for the discovery of many antimicrobial anti-malaria agent. Nowadays many research has been going on, particularly in India. Then we have digitalis, from we obtain digoxin, digoxin, which is used as the cardiotonic or used for the cardiovascular disease. Then also you can obtain or we can obtain medicine from the animal sources. The one of the like famous example is the antibiotic peptides, which has been obtained from African clawed frog, and also some of the analgesic compound has been obtained from the Eucardian poison frog, which is called as epitabine. Then microorganism, from microorganism we can also obtain some of the drugs. So one of the famous drug is penicillin, which has been discovered by the Fleming in 1930s, which is antibiotics. And it was said that once this penicillin has been discovered, the life expectancy of the human being has been increased by the 30 years. So it was firstly discovered by the Fleming with the serendipitous discovery. That was not the like targeted discovery of the antibiotics. So after penicillin, many antibiotics like tetracycline and other antibiotics have been discovered. So it was like golden era for the antibiotics once penicillin has been discovered. So from penicillin, it was act as a lead to develop many antibiotics. Then from marine source, lot of drugs have been derived, particularly for the anti-cancer drug, which includes like discolabine, dolastatin, biostatin. So we can see how many, like even with the venom or toxins from like vipers, we can get some of the medicine which is used for the human beings. Like these are the teprotide, which is obtained from the Brazilian vipers and which act as template for the discovery of captopril, which is a antihypertensive agent which is used for the treatment of hypertension. So as we can see how many drugs we can obtain from the natural source, and either as a drugs or either it can act as a template. And these are the some of the drugs, particularly for the anti-cancer drugs, which can be obtained from the natural source. We know that Taxol, doxorubicin, topetocan, vincristine, vinblastine. These all are the anti-cancer agent or anti-cancer drug. Also, we have enthamycin and etoposide, harmine. These all are the mostly from for the use for the anti-cancer agent. Also, one of the class of the natural product is beta carbolines. So, a lot of research has been done on the beta carbolines. So, mostly beta carbolines act as anti-cancer agent. So, and it acts as the topoisomerase inhibitor. So these beta carbolines itself do not have lot of efficacy, too potent for the anti-cancer activity, but these beta carbolines molecules are acting as the template for the discovery of many beta carbolines derivatives, which will act as the potent anti-cancer agent. So these are the beta carbolin derivatives which has IC50 in the range of micromolar range. So these are the some of the good example of natural based anti-cancer agent. Then also we have benzimidazole based beta carbolin drugs which has been published. It's not drug, it's like agent, this can be act as a template for the discovery of new molecules. Then also we have beta carbolin bisindol type of molecules which have the efficacy as anti-cancer agent. Then there are many more like beta carbolin chalconate derivatives and 1,2,3 triazole beta carbolin derivatives, and also cinnamate derivatives, and many derivatives are then. So these beta carbolins are from the natural cells and it act as the template to the discovery of new anti-cancer agent. So thank you. So we can see how many drugs we can identify from the even one beta carbolin molecules. Any question I would like to answer?
Thank you so much, sir. It was a wonderful session. If anybody from the audiences wants to ask any questions, then you can.
Thank you, sir, for your wonderful lecture. You have very basically explained all the things, and I just want to know that what is the current clinical status of these herbal drugs, sir? As mode of action of many of the herbal drugs is not clear. So, what is the clinical relevancy and what is the current clinical status of these herbal drugs nowadays?
So, as you know, herbal drugs have lot of potential to act as a medicine, but the problem with the herbal medicine is we use medicine as a whole, as a herbal product. So we are not using from herbal medicine we are not able to identify which particular molecules or which particular moiety is responsible for the activity of that particular medicine or particular disease. That's why we are seeing that lot of hurdle because if you are going to file a patent or if you are going to claim this is as a drug, then you have to tell that which particular agent or which particular moiety is responsible for the particular disease. That's why in India, mostly in Indian system of medicine like Unani, Ayurveda, they have their medicine, but they don't have their patent or their claim as a what particular moiety is responsible for the that particular effect. Although we have medicine, but we are not able to tell that this particular effect is due to this compound. That's why we are facing the challenge with the herbal medicine.
Okay, sir. And sir, what is the criteria for selection of human dose for these herbal drugs?
Can you repeat question?
Sir, what is the criteria to select human dose for herbal drugs?
So for selection of any dose, previously you should have the animal dose. You should have able to identify how much animal dose is required. for that particular human dose. You can identify once you have all the parameters from the preclinical study. Then only you can go for the higher animals like horse, pig, chimpanzee. Then it come to the human dose. You cannot identify by calculating itself that this calculation will be the okay for the human dose.
Okay, thank you, sir. Thank you.
Any other questions from the audiences? Thank you, sir, for clearing our doubts. There are no more questions. We can give a big round of applause to sir. Thank you so much, sir.
Thank you. It was my pleasure to present my work here. Thank you.
Thank you, sir. Please have patience with them. Okay.
Hello, good morning, sir. I am audible to you, sir?
Yeah, you're audible now.
Okay, sir. Welcome, sir.
Mhm.
Now we moving on to the next online session by Dr. Kaushik sir. Sir is currently working as a senior scientist at Schrodinger, Bangalore. He received his PhD in chemistry in 2020 from Stony Brook University, New York, USA. Also, sir has more than 10 publications till a date. So let's welcome Dr. Kaushik sir for their scientific talk. Sir, the now mic is yours.
Okay, thank you very much. So I'll be providing all of you a demo session on how to use the software for docking calculations and homology modeling as requested by Dr. Sharma sir. So I'll start with my screen share and then we'll continue after that. So I hope all of you can see my screen right now. Can you please confirm?
Yes, sir. Yes, sir.
Okay, perfect. Right. Yeah, so if you look at the software, for those who are completely new to this, it's a very simple interface where you have a few options on the top left here, and you have this black area which is called the workspace, where any molecule that you're going to look at, visualize it, everything will be visible for you in this particular region. We have a few other options as well. We're going to explore those options one by one as we go along with the demo sessions. So there are a couple of things that you have to keep in mind when you're trying to do this before you start working on any project. The first thing is that you have to define where you want to save the files for your particular project. So you go to File, click on Change Working Directory. Then after that, you can try to choose any folder of your interest. So I'm going to create a new folder for this particular session, and then we can continue after that. So now that I have defined where I want to save the files, the next thing is I have to give the name of the project itself. So I'll go to File, Save Project As, and then I'm going to say docking demo for now. So when you're trying to use this particular package, there are quite a few options that you have available. We'll explore each of those options one by one as we look at different proteins and molecules. So simplest thing that you can do is, before we get to protein-sized molecules, you can just try to look at simple small molecules. So you can go to Edit, and I can go to a 2D sketcher. And once I have gone here, it's going to open up a panel where I can sketch simple molecules. So for example, you can see that it has some basic elements on the left side. You also have like a single bond, double bond, all of these options available for you here. And you have like simple rings that are available also here. So I can add a benzene here. So click on the benzene group here, and then just click on this white area. And you can see there's a benzene that appears on the screen. And I can add maybe a hydroxyl group to this benzene. So I'll just click on the single bond option, put my cursor carefully anywhere on this benzene here. You can see there's a sphere that appears, circle that appears on this atom. So if I click on this, now you can see there's a methyl group that is added. So to replace this particular carbon with a oxygen to make it hydroxyl. So click on O option here and click on the circle here, and I get this hydroxyl. And click on Save as New, and I can save this as phenol molecule. Right? So once I do this, now you can see I have a phenol molecule on my screen here. And you can also see the name of the molecule they have given in the workspace navigator. This is a workspace where you visualize all the molecules. They're similar to your workbench in your lab. So in the same way, you have a workspace to see the molecules. So once you have this particular molecule, you can do quite a few things over there. If you go to this structure hierarchy section, you can see the molecules also displayed here. If you click on the brush option next to this, you can try to change the way the molecule appears on your screen. By default, you have the wire representation, and you can make it a thin cube. You can see the molecule is slightly bigger. You can make it a thick cube. Or a stick model. Yeah.
Dr. Sorry for interrupting you. Can you please change the background, like workspace
So,
background as a white?
I'll do that. Yeah,
Yes.
I can do that. Just hold on. Yeah. And you can also make it like a sphere representation where you have the molecule shown like this. So let me just make it white. Right? Right. So, all right. So you can change the way the molecule appears, and you can also like rotate your molecule. To do that, you have you can either use keyboard shortcuts or you can use a mouse also. So I just wanted to show you a couple of options that is all possible. You can even like zoom in or zoom out. So you can do both of these things with the interface. Right. So, all right. So you can also like do much more fancy stuff with these molecules, but in addition to 2D builder, you can also use like something 3D builder, where you have a build option. This will build directly in the 3D itself for you. So you can go to build, and I can try to use the 3D builder option. I can create a new molecule, and I'll give it as a temporary name here. I can go to add fragments. And if I click on, you can again see like some basic fragments that are available. If I click on benzene, the benzene appears directly on the screen here. And you can click on some particular atom that you want to change. Right? So I can click on this atom, and I can change it to a hydroxyl. You can see that you have a hydroxyl. Right? So you can do these changes very easily with the software. You can also do a lot more things with 3D Builder. For example, if I click on this plus option, I can create a new structure here. I'll try to build a DNA molecule. So if I click on the more option, you have like plenty of pre-built fragments in the software. You have lots of organic fragments that you can add. You also have lots of heterocycles that you can add. And if you click on any one of them, it's going to add them directly to your workspace. In this case, I want to build, you also have lots of diverse fragments that you can add, methyl, ethyl, propyl, all of these different molecules. You can see so many of them are inbuilt in the software. You don't have to sketch any one of them manually. You also have DNA options. If I just do this DNA, you can see there are base pairs, AT, TA, CG, and GC. And DNA is a chain of these base pairs. So I can click on this move selection for chaining option. I can select like one by one molecules, these base pairs at random for now. But if you have any specific sequence you want to use, you can use that sequence itself. So you can just keep building this. And now that you have built it, so you can try to rotate this molecule again and get a view of it. Right. And I can select all of these atoms, and then I can try to say like, I want to make it into a ribbon form. So with a few clicks, I was able to get this ribbon for the DNA. You only get one strand here. You can also get other strand that's not difficult at all. So you can see that in a few simple clicks, I was able to build a DNA as big as this. You can do the same thing for peptide and other type of proteins as well with this particular build option. Another thing that you can do with a build tool is that if you go to, let's take the phenol itself, and we'll try to change some of the group of the phenol. Now I can try to select, like, let's say I select this atom here. I want to replace this atom with some of the group. I can go to build. I can click on create enumerated entries. And once I do that, it's going to open some default functional groups for you. You can see in this particular case, it's going to use the E molecules R group, where you have around 2098 fragments inbuilt in the software. And if you click on the dropdown, you can see you can try to replace the R group library by a different type of R group library. You can choose like diverse R groups or solving R groups or ring decorations, things like that. You can see the number of fragments that are present in each one of them. 43, 33, 24, so on and so forth. You also have R groups to create specific interactions if you're interested in those groups. For now, for the sake of this demo session, I'll just choose diverse R groups, and I'll run this calculation. And you can see that within a few seconds, I was able to generate all of these molecules that have been derivatives of the primary molecule. You have a methane, a methyl group added to it. You can see this is a propyl group added to it. So you can simply sketch these molecules without any difficulty in one single click. Or you can load all of them in one single go also. So you can see all the different fragments have been added to this particular phenol with a couple of clicks. You don't have to sketch anything manually. So this is all well and good for like small molecules, but when you're trying to work with proteins, especially in this case, we're going to do a demonstration on protein ligand complexes. So you can sketch your ligands with the 2D sketcher or the 3D builder, but proteins are like much more complex than your small molecules. So in those cases, most of the time, you want to work with proteins that are deposited in the protein data bank. So in this particular case, I'm going to show you how you can load a protein from the PDB directly. So I'll go to File. You have an option called Get PDB. Every protein in the PDB has a unique ID associated with it. So in this case, I'll try to use a protein with the ID 6B2Q. And I'll try to load a specific chain of the protein chain B and click on Download. So I have this protein, and with proteins, the problem is that you have so many lines present in them, so many atoms present in them. So it's like you can't really make sense of how this protein looks like. So there are a few things you can do to try to change the way the molecule appears on your screen. So you can click on the protein option here and click on this brush option here and click on ribbon. This will display for you the alpha helices and the beta strands on your protein. And now you can see that these coils that you're looking at are the helices, and this arrows that you're looking at are your beta strands. This makes it much more easier to understand how your protein structure looks like. Most of them have helices and strands, and in between you'll have loops between those secondary structure components. This particular protein also has a ligand bound to it. If you want to look at where that ligand is, you can just click on the ligand option here and click on this CPK form. And now you can see where the ligand is in the protein molecule. In addition to this, you can also try to look at the surface for the protein. If you click on the protein option, click on the surface option, you can look at how the protein binding pocket looks like. So you can see this is a protein molecule. That's how the surface looks like. And there is this binding pocket here, and the molecule goes and occupies this binding pocket on the protein. Just hold on. I'll try to change my mouse settings so I can try to rotate it better way. There you go. So you can see in this entire protein, you have like various sort of small grooves here. But the biggest groove you can observe is the one where the molecule binds. And so typically, when you're trying to design your own molecules for a given protein to inhibit it, you want to make sure that your molecules can occupy this deep binding pocket. So just to show you how this molecule, the binding pocket looks like, I'll try to change the representation to a simple ball and stick. And if you look at this particular region and the binding pocket, you can see that it is quite narrow and it's also like a bit deep. So that's what you want to like make sure that you are able to fit a molecule in the binding pocket. So before you can actually use this particular structure from the PDB for your particular docking calculations, you have to prepare the protein. So we have a few tools that you can access in the software, like quite a few of them. If you go to the task option on the top right, so I've seen you so far, like, the workspace navigator. I've shown you the molecules in the workspace. I've shown you how you can change the representation for the molecules by clicking on a few options here. I've shown you the build tool. I have shown you the 2D sketcher as well. Now I'm going to show you this task option on the top right, where if you go here, you can look for various tasks that you can perform. You can do things like ligand preparation, that is essential before you go for docking calculations. You can prepare your protein. You can analyze your structures of the molecules that you're looking at. You can go for virtual screening. You can go for lead optimization. You can go for designing like even MD simulations. You can go for quantum mechanics calculations. You can even design proteins or vaccines as well with this biology section here. You can even design polymers with material section here as well. So The software is capable of doing lots of different calculations. To go through all of these ones will take me around easily a whole week if I want to explore all the tools that are present in each section. For example, if I want to look at protein preparation tools, if I click on this section here, you'll see so many other tools available for you within this interface. For now, I want to fix some aspects of the protein. So you have to run this protein preparation workflow. Because I only have 1 hour, I can't really go into details as to what exactly this particular tool does. I'll give you a brief overview as to what all things it takes care of in this interface. When you have a protein from the PDB, usually it has some missing components present in it. You have to fix those issues. You also probably want to set your pH for the simulation, because when you do your experiments in the lab, you have to use some buffer to maintain the pH of the environment. So in this case, you can click on this global settings option here and set your pH for this calculation. You can also treat metals. Sometimes a protein can have a metal present in them. They might have ions also. You have to figure out how to treat those metals and ions. You might have missing components. You have to fill those missing components. You can go to more options. You can assign or add hydrogen atoms or replace hydrogen atoms. You can create disulfide bonds. You can create missing loops. You can do all of these fancy things with the panel here. And you can optimize the components they have added, run the calculation, and it's going to prepare your protein very easily. So I'll just run this calculation. And while it is running, I'll try to get some molecules that you can use for the calculation eventually. Right. So I've submitted the calculations. The job is going to run shortly. And once it starts running, I'll show you how to monitor the job status and things like that. So job started, right? And I can close this panel once I've submitted the job. And I can go and check its status by going to the jobs button on top right. You have tasks, and you have jobs here. If you go to jobs, then you click on monitor option, and now you can see that you have a job monitor where you can check what is the status of your current calculation. Right? So you can see the files that are being generated. You can see what the status of calculation is. It's going to populate shortly. And once it is done, you're going to have the output on the left side automatically. So for now, I'll just let the job run in the background. I'll try to show you some other aspects of the software while it is running. You have certain things that you can do. For example, I can take this particular complex and I can try to split it into a protein itself and a ligand itself. I have a protein ligand complex right now. So I'm just going to like take this, like right click on it. Actually, I'll just wait. I'll wait for the job to finish. We'll take a look at the results for it, and then we'll do the next step after that. So just to give you a brief overview of what you're doing. So when you have any docking calculation, there are 4 different stages. The first stage is you have to identify which protein is the one that you want to target. And once you identified that, you have to get the structure for the protein from PDB or from machine learning methods. And then you have to prepare that protein. So preparation of the protein is critical before you go for any calculation. Once you have prepared your protein, you have to identify the binding pocket on the protein where you want to dock your molecules. In this case, I have a ligand bond structure, so I can see that one molecule binds here. Most likely, the other molecules also bind in the same region over there. Once you identify your binding pocket, it's the second stage. Then the third stage would be you have to prepare your ligands that you want to dock and see if they're better than this native molecule. And then eventually, you have to do your docking calculation. So you prepare your protein, you find the binding pocket on the prepared protein, then you prepare your ligands that you want to dock, and you do the docking calculation itself. So once this particular protein preparation is done, I'll show you how to find the binding pocket. Then I'll also show you how to prepare your ligand molecules, and then you can do the docking eventually. It should probably take another 2 or 3 minutes for this job to finish. Okay. Right. So anyways, while it is running, I'll show you a few other options in the software. So let me take these 43 molecules that I have sketched. Right? So this was like simple calculation that I just performed with the enumeration tool. And before I can use them directly in the calculation, I have to prepare these molecules. I'll just prepare them for now. So I'll just take this molecules, I'll go to task, and then I'll go to browse. And just as you have tools for protein preparation, you also have tools for ligand preparation. So I'm going to use these molecules for document calculation. I'll just prepare them for now. I can use the LickPrep tool. And in this particular case, you can see that the methods are a bit different. For protein preparation, you have so many things to take care of in terms of the right bond order for each of the atoms that you have in your molecule and your protein. In this case, for LickPrep, you have to figure out what is the charge going to be for your small molecule at the pH that you have mentioned. So if you remember, like if you change the pH, the charge on your molecules can change. They can either become positively charged or negatively charged depending on what pH you set here. So I'm going to first choose which molecules I want to prepare. I'm going to prepare all these 43 molecules, and I'm going to generate all possible states for them at a pH range of 7 plus or minus 2, which is around neutral pH. You can also generate tautomers for these molecules by checking this option here. And you can also generate stereoisomers for these molecules, R form, S form, and all of that stuff by using these options here. By default, it's going to give you around 32 stereoisomers per ligand. If you have one color center, you get 2 stereoisomers. If you have 2 color centers, you get 4 stereoisomers. If you have 3 centers, you get 8 stereoisomers. If you have 4 centers, you get 16 stereoisomers. If you have 5, you get 32 stereoisomers. If you have 6, you get 64. In most of the time, it's very, very difficult from a synthesis perspective to get only one particular stereoisomer. So in most cases, drug molecules will have maybe 1 or 2 stereoisomers, not more than that. Chiral centers and not more than that. So in this case, I just want to keep things simple for this demo session. I'll just choose only one stereoisomer to be generated, and I'll run this calculation for this setup. So to make this run a bit faster, I'll try to use more CPUs so that I get the results a bit quickly. Right? So I'm preparing these molecules at this particular pH range, generating tautomers, genetic stereoisomers, and once I get the output for these prepared ligands, I'm going to dock these molecules eventually into the protein binding pocket. I'm still waiting for my protein preparation job to complete. Right. If you have any questions on what is covered so far, please let me know. We just have to wait for this section. Actually, in the meanwhile, I was also asked to do a demo session on homology modeling. So for that, I'll try to do it. So let me try to look for a protein sequence. Just hold on. So just as you have the PDB for the protein structures, you have UniProt, where you can get your protein sequences. So it's usually not this slow. Like, unfortunately, my laptop yesterday from water fell on it, so it's not working. So I'm using a temporary laptop right now, which is why everything is like running a bit slow on this particular machine. Otherwise, it's typically like the jobs would be done by now. Okay, all right. So you can search for the protein of your interest, and let me. I'll try to look for, let's say, phosphoglycerate kinase. Once you search for the protein of your interest, you can look at the results for it. And you can see that it tells you the entry ID here. It also tells you what is the name of the entry. We can see the first one is for mouse, second is for humans. You can also see the organism on the right side, which tells you clearly which species the protein belongs to. Let me take this human one, T07205. And if you look at this particular protein, for this one, if you go to structure section, you'll observe that as of now, for this particular protein, I don't think there is any structure available in the protein data bank itself. So in this case, you'll have to build a structure. So the only structure we have right now available for this protein is from the AlphaFold method, which is a machine learning method. So you want to probably like build a structure from the sequence. So you can get the sequence for the protein from this region here, and you can download the sequence as a FASTA format. So you can click on the download option, and you get the sequence available for you in this case over here. So once you get the sequence for this particular protein, then you can go to a tool that can help you build your homology model. So you can go to the task option here and go to browse, and then I can go to biologics. And here you have a tool called multiple sequence viewer / editor. So once this particular panel pops up, I'm waiting for it to come up. There you go. Once it pops up, you can go to the file option. You can paste your sequence, right? I'm going to give it a temporary name for it. So it is the query sequence. Let's just say that. And then you can paste your sequence here. Let me try it again. Yeah, there you go. So you click on, let me delete it and paste it. There you go. All right, and click on add. And so that's going to add your sequence to this particular interface. And once it's added, then we have to search for suitable templates in the protein data bank using which you can build this structure for this protein. So you got the sequence, and then once you have done that, then you can go to this other task option, and you have an option called build homology model on this right side. And once you click on it, it's going to open a panel for doing homology modeling also. So I apologize again. This is a new laptop. I just opened it like half an hour ago because my personal laptop has a repair issue right now. There you go. Right. So I got the query sequence here, and then after I can try to find the template structure and click on the find option available here. And I can try to search across the PDB to find a protein that has a similar sequence to this, along with the structure for it. So it's going to do the search right now. Okay, I need to change a couple of settings before the search happens. Let me just do that. All right. So if I click on find right now, it's going to start looking for suitable templates from the PDB, and once it finds them, then it's going to ask us to choose the right template for it. So while this is running, I'll just like minimize this interface, and I'll go back to the Maestro itself, and I hope the jobs are completed by now. Nope, they're still running. This will take some time then. If you have any questions in the meanwhile, let me know. We just have to wait for these jobs to complete.
If anybody from the audiences want to ask questions, then you can ask freely.
Students can perform covalent docking.
Yeah, you can do covalent docking for India. There's a specific panel for that. So if you go to task and browse, and if you go to the section for Glide, which is a tool for docking calculations, you can see there's a panel for covalent docking. Right, so you can do that. Yeah, and if you look at it, you can, just to give you a small hint on what type of things it can do. So you can look at the reaction types, and you can choose any of these reaction types available for your Michael addition or nucleophilic addition or Bonica substitution. All of these things you can do without any difficulty. Yeah. You can also create your own custom reaction as well if you want to form a covalent bond between the ligand and the protein. Any other questions?
Sir, if we have a compound and want to know its synthetic step of any compound, so is it possible to solution that we can find the synthetic step on?
Can someone repeat the question? The voice was not clear from far.
Sir, synthetic step of any compound, if we have a compound and want to know how it can be synthesized.
You have a compound, and what do you want to do with it?
If this molecule, if I have a drug paracetamol and want to know the synthetic step. So,
Sorry, the voice is echoing quite a bit. Can someone like try to clarify it?
Sir, suppose we have a paracetamol molecule and want to know the synthetic step, how it can be synthesized. So,
Okay, yeah, I can do that. So just let me just show you for let's say one of these molecules here. So with a bit of a complication. Right, you have this molecule. It's not the same as paracetamol, but it's like slightly bigger than that. So just hold on. Someone. Okay. So if you go to task, and if you go to browse, and if you go to the applications again, there is a tool called Pathfinder that tells you how you can synthesize a molecule. So job is completed, one of them. That's good. Just a minute. You still hear me? Am I audible?
Yes, sir, you are audible to us.
Hello?
Yes, sir, you are audible.
Can someone confirm if you can hear me?
Yes, sir, we can hear you, sir.
Yes, sir, you are audible.
Am I audible?
Yes, sir.
Hello?
Yes, sir, you are audible.
Let me try. I'll try to rejoin the call. Just hold on. Yeah. Can you hear me now?
Yes, sir.
Okay, perfect. Thank you. All right.
Okay, sir.
So, all right. So I have the output for the calculations right now. And you can see I have the liquid output where the molecules are prepared. And you can see that right now there will be that they're all like, probably the geometry and orientation will be perfect for them right now once they have been prepared. So okay, that is done. And in the meanwhile, I'll also show you the options for multiple sequence viewer that we were trying to find the templates for the protein, right? So just hold on. Just hold on. So for the homology modeling, it has like searched the PDB and it gave me like different PDB IDs that could be possible templates for this protein. And so if you look at the results for it, you can see it shows you all of these PDB IDs on in the first column. And you can look at what is the similarity or identity percentage between the different PDB IDs and your query sequence. So in this case, I chose this particular PDB ID as the template and I imported it into the software. So you can see that it says 2ZGB_A for this particular protein which we're going to use as template. Once we have done that, click on Run Alignment, and this is going to align the two sequences onto each other. And once this alignment is done, then you can click on this Choose option, where you can choose the ligand that you want to use. So it's always good to start with a bound protein complex for drug discovery compared to unbound protein ligand complex. So in this case, 2ZGB has a ligand bound to it. You're going to use the same ligand to build our structure for this query sequence as well. And then eventually you can click on the view settings option and you can try to choose whether you want a knowledge-based method or a energy-based method. Knowledge-based method is good when you have a very high sequence identity, like this case. If you have like a low sequence identity, like around 50-60%, then you can go for energy-based method and try to get your structures. So click on OK right now. It's going to start. That's the settings that we have here for this particular protein. And the job name has to be usually given. It's up to you whether you want to give it or not. But I recommend you to give this job name. So I'm going to call this as PGK2, and then I can try to run this calculation to build this homology model structure. So essentially the idea is that you take your sequence for your protein for which there's no structure, you get the sequence from UniProt, and then you try to find a template in the PDB by clicking on this find option here. And then once you have got a suitable template, then you run the alignment, you choose the ligands that you want to add, and you click on the run calculation. It's going to start building this particular model for you. So this is going to start preparing the homology model for us. And while this job is running, we will try and take a look at the retrosynthetic pathway analysis that was asked by one of you. So for this particular calculation, I'll just try to show you one of the ligand molecules. So let me take one that we have already generated from this list. So I have this molecule, and I can figure out what is the right synthetic pathway going to be for this molecule. I can go to task. I can go to the browse option, and you have a tool called Pathfinder on the left side. So if you open this, you have a tool called reaction-based enumeration. So you can just click on that particular panel. And once it pops up, then I'll show you how to use this particular panel. If you have any other questions, please let me know so I can try to address them in the remaining half an hour that I have. It is usually not this slow at all. It's an old laptop, so and I just opened it like 20 minutes ago, so I don't know what's happening here. By now, like most of the job should be done, and even the demo would have been over by now. Thank you for being patient.
Hello, Kaushik sir.
Yeah, can you hear me?
Sir, how can we perform DNA docking or protein docking? So, if the software is not loading the thing, hello. Hello, Kaushik sir.
Yeah, can you hear me?
Yes, sir. We can hear.
Okay. Just
Sir,
hold on, right?
I'm
Yeah.
asking about DNA docking or protein docking. So just give the information. If the software is not loading the things, so just give the information. What is the difference in ligand docking or DNA docking, and what the tools we can use through the Schrodinger software?
Yeah. So the difference between small molecule docking and protein docking or DNA docking is that the binding pockets of protein DNA complexes or protein RNA complexes or even DNA itself typically has like it's a shallow binding pocket. It's not a deep line pocket like the way you have seen for the small molecules. So because of that, you have to use a different approach for the docking calculation. Also, the size of this molecule is very big. In the case of small molecules, because they are smaller, because they are like have few atoms, you can go for flexible docking of the ligand, and you can have a rigid protein to the docking calculation. But when you go for protein docking, both the protein and the ligand are going to be like big, so you can't like try to you can't do the regular docking itself. You have to have a different approach for it. So there's a tool called a Piper, which is specifically for protein docking or protein DNA docking. If you want to dock like a small molecule to a DNA, it can still be done. We have a new interface for RNA docking as well. DNA docking already supported long time ago, and we have fixed some of the issues with the accuracy of the tools for DNA docking and RNA docking, small molecule DNA, small molecule RNA. But for protein, the Piper tool is pretty robust. You can do that calculation. You can try to get the information very easily. Just because the size of the molecule is very different, you have to use a different approach altogether compared to small molecule docking. Anyway, so in this case, this is a panel for the retrosynthetic analysis. So I'm going to load the molecule that I have in the workspace. So I have this molecule, and I can click on like how many depth you want, like how many different levels you want to have in your reaction pathway. If you click on the pathway here, generate pathways, it's going to run the calculation right now, and it's going to show you what are the possible synthetic pathways for this particular molecule. Right? This usually takes like 1 or 2 seconds, but it's a new laptop, so let's see. And while this is running, let me see. Well, yeah, even the main maestro is not responding. Okay. Any other questions in the meanwhile?
Hello.
Yeah,
Sir, this
yes,
is
ma'am.
Sir, can be done DFT study by Schrodinger?
Yeah, you can do DFT studies. So yeah, there is we have a model called Jaguar that can do even single point calculations, even like QM optimization actually can be done. So I'll just show you once it responds. Okay, now it's responding. Great. Fine. So if you look at let me just try to show you the options here. Let's just wait for this to finish, and once it is done, then we'll go to that DFT part. I'll show you what you can do with it. So anything you want to do with your structures, you can do it. That's the simplest answer I can give you. So you have to reduce a problem to a molecule level problem, and you can try to use the tools that we have right now. We have three main aspects. We have small molecule drug discovery. We have biologics, which can be helpful for protein design. And the third thing that we have is material science. So material science has tools for building polymers, nanostructures, nanotubes, all of those things. You can also try to model like diodes and electronic devices with material science suite. Then you have biologics where you can design like antibodies and vaccines, things like that. And you have a small molecule where you can find out the simple inorganic molecules for trying to inhibit a particular protein. So if you look at this, is the output for the retrosynthetic analysis, where you can tell you that given a molecule, what are different reactions you can use for getting those molecules to be present. And let's say your lab is like you're very good at doing one of these reactions, you have all the reagents available for it. If you go to define reactants option in this particular panel itself, you can try to choose which particular reactant you want to use for generating products that can reproduce by a given reaction. So just wait.
Sir, Kaushik sir.
Yeah, yeah.
Sir, as we see that the starting material is also so big, so that is not easily available, I think, in market. So we further need to derive a synthetic step for these compounds, as you showing in the
yeah. So if you want to further break it down, you can go for like so. So actually, you can mention like how many steps you want in the reaction. If you want to break it down further into smaller steps, you can go to like, right now it just shows like one single step through which you can combine this particular to get this molecule, right? But you can go to like 2 steps or 3 steps. And if you go to 2 or 3 steps, then you're going to get even sub-fragments that you can use to build your molecule, right? So I'll just show you the option that you can use to get a multi-step reaction pathway as well. So, but for now, let's just hope it responds at some point of time. The water spilled on my laptop like last night at 9 p.m. So today morning I had to make some computer arrangement to do this demo session. Very sorry for that. Okay, one of the jobs is completed. I don't know which one it is, but we'll see. Okay, so here you can try to define the reactants for your reaction. So let's say you want to use this particular reaction and you want to get all the products for this particular reaction. So you can see that you have some groups that are available. Reactant 1 is defined, reactant 2 is defined, and in this case, reactant 2 has it's going to use organostanins. So there are around 478 different fragments available in this reactant 2 library. And for reactant 1, you can see it has around 17 lakh halides, aryl, and vinyl groups over here. And you can, if you combine all of these into one single place, you're going to get lots and lots of different products that can be made from this reaction. And you can limit the number of products you want to have. But going back to your earlier question on can you increase the depth of this reactions to multi-stage, you can set this option here from max step 1 to 2 or 3. And if you do like 2 or 3, you're going to break it down into even sub components for making this particular reaction. Since it is hanging up quite a bit, I won't go into how to do that particular thing. There was a question on whether you can do QM calculations or not. So if you go to task and if you go to the browse option, and if you go to this section for Jaguar, you can see that you can do single point calculations, you can do optimization calculations, and if you go to, let's say, optimization, you can it's taking some time to load. The protein is prepared. That's a good thing. So at least I can complete the docking demo for you once it responds. Right, so you can set your level of theory here. These are all DFT-based methods. So you can choose any one of these, MO5 or MO62X, whichever one you want to use for the level of theory. And you can define your basis set also. There are quite a few options available in this case, right? So once you have done that, then you can also go and choose how many iterations you want for the SCF cycle. You can look at the optimization properties that you want to choose. You can also try to measure some properties where you can just choose if you want to get the vibration figures, you can select this option here. Or if you want to get like charges on your atoms, you can try to choose this ESP option. You can get your surfaces, you can do NBO analysis. You can also try to calculate HOMO-LUMO and all of those things as well. So you can do that stuff with the Jaguar module in Schrodinger. Right? So that is with respect to that question. So here I have the prepared protein. And so once I have this prepared protein, so I have done 2 stages right now. I have prepared my ligands, I have prepared my protein as well. And the next stage is I want to define the binding pockets. I can see there's a ligand bound here. I want to tell the software that is where I want to choose, that is what I want to choose as a binding pocket. So to do this, there are 2 steps that you have to perform. So if I just click on this preset, apply custom preset option, it's going to zoom into that ligand in my complex. And once it is done, then you can go to task and you go to browse, and we have a tool called Glide that does all the docking calculations for you. So if you go to Glide, you have an option called receptor grid generation. And once you open this particular panel, the first option that you get is you have to define your receptor. So if you're structured in the workspace, this is the workspace. It's a receptor plus a ligand. So I have a receptor and a ligand. It says you must identify the ligand molecule so that it can be excluded from the grid generation. So this ligand has to be removed so that you can put your molecule. So this step will recognize this ligand molecule. So I'm just going to click on any atom of the ligand. It's going to identify all the atoms of the ligand for me once I do this. So I can see that now all the atoms of the ligand have been green colored cubes. And if I zoom out from the protein, you will see there's a pink box around this region that is the binding pocket for this particular protein that I'm defining right now. So I can just run this calculation, and that will define a binding pocket for the protein. Once it is done, I can just take some of my molecules that I have prepared, and I can dock them into this binding pocket. So you have to prepare your protein, find the binding pocket using this grid generation option, and then the ligand will be removed that is present already, and we can dock our own molecules into the protein eventually. So this should ideally take not more than 2 or 3 minutes, but since it's been running slow, I hope it doesn't take longer than 5 minutes. So if you have any other questions, let me know. And otherwise, I'll just show you like other things that you can do with the software. If you go to like browse and biologics, you can try to like model antibodies. Yes.
Sir, a question. How we check which pocket is right for my drug?
So it's not, you can't say the binding pocket is not for your drug molecule, it's for your protein. So proteins can have different binding pockets. You have to find the right binding pocket for a given protein. In most cases, like if you have a ligand bond structure available from the PDB, that is the most likely binding pocket for you already. If you don't have a ligand bond structure, you have to do either homology modeling to get a ligand bond structure in some way or other. But if you're left with no other option to get your ligand bond structure, then you can use, there are two options then in that scenario for you. One is that you can look at the literature and identify which amino acids are critical for the activity of the protein. If you can find that information from the literature, then you can be sure that that is your binding pocket. If you don't even have that information, there's a tool called sitemap in Schrodinger, where if you go and use this particular tool called sitemap, that will predict the binding pockets for you on the protein. It's going to rank order which one is likely binding pocket, and it tells you a property called D score, which stands for druggability score. It tells you if a molecule can, if a pocket, binding pocket can have a drug molecule or not. So if the D score is more than 1, that means the binding pocket is druggable, and then you can try to choose that and define that as your binding pocket for your molecules. Okay, right. So if you go to biologics, you can see that there's a tool called protein-protein docking, specifically for docking a protein to a protein. You can also use this for protein DNA docking or protein RNA docking as well. You have tools for trying to do mutations to your proteins. You can engineer your protein. You can analyze your protein as well. You can predict your structure also for your protein. And it's similar to the one that we had done for protein homology model as well. If you go to materials, you can try to build so many different things in the materials section. You can go to structure builders, where you can build like complex formulations for your molecules. You have, you can build like polymers. You can build disorder systems. You can also build like nanostructures, nanoparticles, and things like that. You can try to build meta-complexes as well. So all of this can be done with the material science options that we have available. And once you have built those systems, then you have to run either MD simulations or you have to go for quantum mechanics, depending on what type of machine you're trying to make over there. So the grid is still being prepared. Once it is done, then you can go for the docking calculation. I still have 10 minutes left for the demo session. So I'll just wait for it to finish, and then we can cover the other aspects. In the meanwhile, you can also do like induced fit docking. You can go for pharmacophore modeling as well. You can do shape-based screening of the molecules. You can also go for tools that can optimize your molecules. You can predict ADV properties also in this particular interface. So for now, the grid is being prepared. And once it is done, I'll just dock maybe 4 to 5 molecules, not more than that, because the way it is happening right now, it's very, very slow. So I just want to show you like docking of 2 or 3 molecules and not more than that. Any other questions? Please let me know. Just know that, any molecule that you have, if you can reduce your probability at structural level, you can use these tools for that calculation. We have covered briefly on covalent docking, reaction-based enumeration, retrosynthetic analysis. I've shown you homology modeling. I've shown you how you can do even, like, I showed you the panel for QM calculations as well, DFT calculations. You can also try to model like antibodies and things like that. So the software can do lots and lots of different things for you.
If anybody has questions, you can ask now.
Hello.
Yes, ma'am.
Yes, sir. Sir, how can we validate our docked pose through the software, through this software?
So you don't, well, validation is a good aspect of it, right? So the software has been used by a lot of people over the last 20, 30 years. So there are plenty of papers that have been published along with it. So that's itself is a validation that the software is good enough. If you want to do your own validation, so typically what people do is if you have a protein ligand complex, they take the bond molecule itself, they do the docking for the bond molecule. So the idea is that, you know, from this, because the structure is from the PDB, you know exactly how the ligand is supposed to bind in the binding pocket. And if you take the same molecule and do the docking for the molecule, you should get the same pose as what you get from the experiment. If those two are close to each other, that means the software is giving you the pose that is very, very similar to what the experiment is giving, which indicates that it is accurate enough to reproduce experimental binding pose. That is the best way to validate your docking calculations, that you're getting the right results. And this has been done plenty of times for Glide, for Schrodinger Maestro module. So you don't have to worry about validating your results. If you just cite the software, people will accept the results automatically. But you can always do a quick cross-check if you're getting the right results or not by just taking the bond molecule and running a docking calculation on it and see if you get the same pose as experimental binding pose. I hope that clarifies your query. You can also go and try to add decoys and try to see if your software can capture the active molecules in the decoys. So that can be done. You also have tools for enrichment calculations that can help you with that type of analysis. So my grid is prepared. So I want to do the basic docking calculations for, let's say I'll just take this like 3 molecules, not more than that. So I'll select this 3 molecules. And so I can go to the panel for ligand docking. So once I have open this thing, so I have to mention the grid file that I have generated. This tells you the binding pocket information. You click on the browse option, and all the jobs that I have run, now you can see that you have an option called enumerate, where I have generated those 43 derivatives. I have this lickprep output calculations. I have this homology modeling output that is being run still. It's not yet done. I have protein preparation folder. I also have a grid generation folder. I can go to this grid folder, and I can select the file that has the information on the grid, which is this file here. I'll just open it. That is a binding pocket. And I want to dock these 3 molecules into the binding pockets. I'll just choose those 3 itself by clicking on this drop down. I could have chosen all 69 of them if I had time, but right now I don't. So I'll just like run this calculation quickly, and hopefully we'll get the results in the next 5 minutes. Let me see. So go to settings, and I choose 3 and run the calculation. Hopefully it finishes. Usually it takes like 10 seconds to dock every molecule. I'm expecting it to not take more than 1 minute for this one. So let's see. If you have any questions in the meanwhile, we can take up. I think, yeah, 12:25 is when I'm going to end this session.
Thank you for this wonderful session, sir.
Yeah. Docking is completed. If you can, I can just take 2 more minutes maybe and show you the results for it. Probably if you have time. Otherwise, I can end this. Well, it's still running actually. So yeah. Yeah, if you want me to conclude, I can stop it right now.
You can proceed.
Okay, thank you. Yeah, I'll just kill this homology modeling job, so at least the docking one will run faster. because anyways, it won't be done in the next 2-3 minutes. Just to add in the homology modeling part, once you get your structure, you have to check the quality of the structure. There are tools to do that. So we can look into it in the future sessions that are possible, but not now. Anyway, once the docking is done, if you go to table here, you'll get the docking score, which is typically used for finding the best molecules from the ones that you have screened. And you also have tools like ligand interaction diagram, which tells you the interaction between ligand and protein in 2D. And you also have a interaction toggle in the bottom right that tells you the interactions in 3D itself. So for example, if I turn this on right now, and if I look at the binding pocket, you can probably see that it's going to display this dotted yellow lines that tells you which atoms are interacting between the protein and the ligand. So essentially, the analysis for docking would be like, you take your molecules that you have docked, and then once you get the results, you have to see which one has more interactions and what type of interactions they have. So that is the main part of docking analysis. I think the job will take some time to run. So since I only have 1 or 2 minutes left, I think I'll stop the presentation here. I'm very, very sorry that I couldn't use my own laptop for this presentation since I had a water spill last night. But I hope you got the general gist of the overview of the software as to what it can do. And if you have any questions, please let me know. Otherwise, I think we can come to the demo session right now. Thank you.
There are no more questions, sir. Thank you for this wonderful session. Thank you, sir.
Thank you, ma'am.